Next, we'll move on to just some tree diseases that are fairly common. First, one I guess a lot of you guys have seen, but that's Diplodia tip blight. More recent name for it is Chiropsis tip blight, but Diplodia, people still know what you're talking about with that. Diplodia tip blight tends to, on the needles, will affect this year's needles primarily, which we can see on the picture there. And then also, when you look at the cones that are formed, there may be a bunch of these small black pimple-looking things, just these black dots. And here we have a close-up of these. I like to call them pimples, but they are pycnidia. And so those are fruiting bodies of the fungi, of the Diplodia fungus, that are erupting out of the bottom or out of those pine cones. And then each one of those little pimples will have dozens, if not hundreds, of spores inside of it. To control Diplodia tip blight, this is one of those diseases, as with a lot of our fungal diseases, favored by rain and high humidity. And so if you're able to decrease humidity in any ways, such as pruning or making sure that things are properly spaced, that should really cut down on the amount of Diplodia tip blight that you get. Also, if you are seeing a lot of infected cones on the ground, you may want to go ahead and clean those up too, since the spores will overwinter on the infected needles, but also on the infected cones. And while in a single year, Diplodia tip blight will not be a major harm to any of your trees, if you continuously have this disease, eventually it's going to severely impact your tree. As you can see from the picture that we have, this tree is not long for the world when this picture was taken, and it's now gone. But after a few years of repeated infection, it will eventually kill the tree. So if you do have a history of Diplodia tip blight, we would recommend doing, this is one of those when we would recommend doing a protective fungicide spray. And so this is a fungicide spray at two different times. First, you'll want to do about the third week of April, and so right as the new needles are starting to emerge, and then you'll want to do that second application about the first week of May, or when those needles are about 50% elongated. And so those two applications really do a good job of controlling Diplodia tip blight. If you don't want to spray a fungicide, some of our copper products, such as Bordeaux's mixture, are very effective at controlling this one too. Next, we'll look at some of our needle cast diseases. So again, they affect a lot of different types of conifers. But these needle cast diseases, instead of affecting this year's growth, they tend to infect second and third year old, or 2- and 3-year-old needles. And so as we can see on the pictures, especially the picture on the bottom left, and then also the one on the right, is that this year's growth is looking pretty healthy. But it's as we go back to 2-year-old, 3-year-old needles, that's when we start to see the injury occur. And if you would look at the needles underneath a magnifying glass or a microscope, similar to Diplodia, you would start to see a whole bunch of little black pimple-looking things that are kind of erupting out of the needles. And again, those are the fruiting bodies of the fungus. So we have two main needle cast diseases that are in Nebraska. The one that a lot of people are familiar with is Rhizosphaera. Rhizosphaera has been around for a long time. People know what it looks like. But about 10, 15 years ago, we started to see another needle cast appear, primarily in the Great Lakes region, but it's since spread throughout the country. And this is Stigmina needle cast. And so it's a very similar looking disease, but they are two different fungi. And so you can kind of tell them apart if you would happen to look at them underneath the scope. Rhizosphaera, those fruiting bodies tend to be pretty spherical, nice and round. They almost just look like a basketball that's coming out of the needles. We also tend, with Rhizosphaera, we also tend to see the needle, or see those fruiting bodies are very much associated with the stomata. And so you will see them appear in lines on the bottom of the needle where the needle stomata are. Compare that to stigmina, and as you can see on the picture here, for stigmina, those fruiting bodies they're not near as spherical, not near as round. Underneath the microscope, they almost look like a dead dried spider that's flipped on its back. And so you'll just see a bunch of kind of hairs that are coming up out of it. But that is what a stigmina tends to look like underneath the scope. Both of these infect second and third-year-old needles, but unlike Rhizosphaera, stigmina doesn't appear as uniform on the needles as Rhizosphaera does. And so stigmina is going to have, those kind of spider-like fruiting bodies, but they won't be near as uniform as you would see with Rhizosphaera. Luckily, management of these two diseases, and I apologize for these slides, there's a lot of words on them, but mainly it's just for your to take home. But management of both these needle casts is pretty much the same. As far as cultural practices that you can do, if you're able to clean up the fallen needles, that's a great way to decrease the amount of inoculum that you have. Also, put some mulch around the base of the tree. Try to make sure there's not a lot of grass that's growing right up next to the base of the tree. If you have grass that's growing up to the base of the tree, that can really increase your humidity levels there as well, which can increase the amount of needle cast. But for both of these, we recommend 2 applications per year. One of the problems is with stigmina, this one, if you have it, it may require indefinite applications of fungicides. So for the next, at least for 3 years, you would need to do a fungicide spray, but research is ongoing if that fungicide spray is needed for 5, 10 years down the road to save that spruce. Rhizosphaera, on the other hand, typically if you do 2 years of a fungicide spray for Rhizosphaera, that will knock it down to a point when your tree will no longer be negatively impacted by the disease. Next is a very common one that I've seen is pine wilt disease. It primarily affects Scotch pines, but now that a lot of the Scotch pines are gone, they're starting to move into some of these other pines that we have. Can affect white Austrian pines. We have seen it on ponderosa pine as well. Typically, pine wilt disease infects older trees that have some other stresses going on. We really don't see it on young trees all that often. So about 10 years old is the youngest trees that we tend to see pine wilt. But we will get this very rapid decline over the course of just a couple of months with this disease. The picture that we have right here, in the spring, that tree was completely green. By fall, it was dead. So just over the course of a couple of months, the tree was completely wiped out. And the causal organism for this disease is the pine wilt nematode, Bursophalanchus xylophilus. But that nematode is actually vectored by a beetle. And so the pine sawyer beetle spreads those nematodes from one tree to the next. And now we're going to get into a little bit of the pine wilt disease cycle. It's a pretty complicated disease cycle, includes 4 different organisms, and so it's a great one that I like to talk about. But so here we have a fairly healthy pine tree, and then we have a pine sawyer beetle that lands on that healthy pine tree. Inside of that pine soil needle, or inside of that pine sawyer beetle, it's going to have some of the nematodes inside of it. As the beetle feeds on the tree, those nematodes will then escape the beetle, and start to get into the tree themselves. If you have a resistant host, those nematodes will then die as soon as they get into the tree. However, if it's a susceptible host, Scotch pine, Austrian pine, something like that, well, now those beetles can, or now those nematodes can start to reproduce and increase in numbers throughout the tree, inside of the tree. And what you might be able to see if you peel back the bark is some of these nice kind of curved galleries underneath the bark caused by the beetle. The other thing that you will see is that when you cut the tree, there might be some kind of blue stain. Looks like the part of the branch has just been stained blue, and that's the blue stain fungus. And the nematodes are fungal feeders. And so as the nematodes reproduce in increased numbers throughout the tree, they are moving this blue stain fungus throughout the tree as well, so that they have something to feed on. And as the tree continues to decline, as the amount of fungus inside of the branches and nematodes both increase, the tree is declining and now is sending out a whole bunch of stress pheromones, which means that the bark beetles, or the Sawyer beetles, are now attracted to these diseased trees. They're going to come and they may come and feed on it. The other thing that they can do is that the pupa that the beetles have laid in the tree, they can eat some, or they can absorb some of the nematodes as well. But then now we have an emerged beetle that has a gut full of nematodes, and it's out searching for another healthy tree. Again, pretty complicated life cycle. It's one of my favorites to talk about. So thank you for indulging me. But as far as management and identification of this disease goes, chemical controls have had kind of mixed results. They tend to be fairly expensive and require repeat applications as well. So removal of diseased trees is one of the best options. But you want to try to remove these diseased trees by the end of April. The bark, or the beetles, emerge as we get into May and into some of the summer months. And so we want to try to get rid of that tree before the beetles have actually emerged. And then that tree should be, the wood should be burned or buried. You don't want to chip it. If you chip it, the nematodes can survive and they can go on to infect something else. As far as sampling the tree for pine wilt nematode, you want to make sure that you get a large enough sample that we can actually test something. Get quite a few samples into the clinic for pine wilt nematode on branches that are about, half an inch in diameter. So some of the smaller branches that may have already fallen to the ground. We're not going to find any nematodes in that sample. So really, we ask that you sample wood, either take a core from the main trunk, or try to collect a branch that's at least 1.5 to 2 inches in diameter and about 6 inches long. And that should be a big enough sample that we can find the nematode if need be.